Tomorrow's therapies will aim to address the significant challenges encountered in oncology. Oncologic diseases are complex, and each patient is unique. The development and regulatory pathways for a treatment sometimes are not sufficiently focused on the unmet needs of the patient population. So, what are the keys to achieving an oncology medicine that puts the patient at the center of care?
Hans Scheurer, thank you for being with us. You are a vice chair of the Weekend Work Group of European Union Cancer Patient Communities. It's a network of leaders from over 20 cancer patient umbrella organizations in Europe. You have been an active member of Myeloma Patients Europe since 2013. You have been a patient yourself, and you are a patient advocate of healthcare projects that support and empower cancer patients and patient group. What are your thoughts about this video, and what about the importance of having personalized treatments?
Thank you. I actually am still a cancer patient, not only have been. Each patient is unique. That's the main message, I think, and I would approach it from two angles, two issues, two challenges maybe. Keep an eye on the individual differences, I think. That's the first one. And what I mean is that we especially look, and also the regulators look at the big data. So the big report with all the data and all the, well, look at what the efficacy of a new treatment is. But a lot of patients don't fit exactly in those big data. And those are exactly the gaps of the knowledge we don't know often. So, for example, multiple myeloma, the former speaker already spoke about that, and multiple myeloma is a bone marrow cancer, medium age 70, overall survival medium about 8 years. But we have a lot of patients that don't fit in that group. Me, myself, for example, I have myeloma for 19 years, and when I was diagnosed in 2005, I was considered to be a high-risk patient. And I recently spoke to someone in the room who said my father had myeloma and he has lived with it 25 years. So there are a lot of patients that don't fit in those general picture. And those people, I think we need to keep an eye on, because especially the patients that don't fit in the big data reports show where the knowledge gaps are and where we need to get to a more targeted approach and precision medicine, I think. The last point I want to make, because I only have 3 minutes, and that's more than enough to make those 2 points, is about PROs. Patient-reported outcomes. That's the other point where you can see that patients are unique. When we speak with regulators, also with pharmaceutical companies, the focus, especially with regulators, is on overall survival, for example. But that's difficult to look only at that kind of endpoints to measure the value of a treatment. Always say when I'm at regulators, but also elsewhere, the proof of the pudding is in the eating. So that's why you need to ask the patients what the value is on their day-to-day. Not only the data where you have your blood counts or other things you normally measure, but the patient lives day by day by day by day with the disease and with the treatment. So you really need to have an eye on the impact of that treatment on the daily life, the quality of life of the patients, to get a real view on the quality of your product, on the value of a treatment. That's the point I want to make, and just because pro-measuring is difficult, it doesn't mean that we should avoid it. Thank you.
Thank you very much, Hans. It's now time for a second video that tackles another challenge. Let's discover it right now.
Every patient has unique needs. To develop new therapeutic solutions that address these needs, we must better engage and involved patients. It is a crucial element in achieving participatory and personalized medicine. So, how can we directly involve patients in the development of new oncology drugs, and what impact can they have?
Dr. Ines Vaz-Luis, hello. Thank you for being with us. You are a medical oncologist and researcher specializing in breast cancer from Gustave Roussy, and you lead a medical scientific program dedicated to post-cancer care. Empowering patients is a real challenge, as we have seen in the video right now. How can we directly involve patients in the development of new oncology drugs?
Okay, so let's start by why, that was already a little bit touched on, and then go to the how question. So we know in medical field and outside of the medical field that when we involve the end users, stakeholders in the construction of products, we'll have better reach, better adoption, better effectiveness. So when we think about why should we involve patients in a two co-creation dynamics, I would say that it's not only for normative reasons, because patients have the right to be involved in projects that are focused on them. It's not only for it's not only for instrumental reasons, so we have the trust of our patients, but it's also for the substantial reasons that our products, our research projects, will be better. And I will give you an example. I'm involved in this European project that is focused on develop a new cellular therapy. And so the focus is try to prove that this new therapy is safe. And what we did, we have a full work package dedicated to patient experience with a task dedicated to co-creation. So since the beginning, we had a stakeholder board with patients, and we trained the patients, and we involved the patients in the protocol construction. And what happened was that we moved from a primary endpoint that was only focused on looking at adverse events measured by the physician to a combined primary endpoint where we have adverse events measured by the physicians, but also PRO CTCAEs. And we also changed the way supportive care was drafted in the protocol to truly create pathways that were adapted to the patient experience. So how do we do this? First, we have to want to do it. Then we have to partner with people that are able to do it in a formal way. And we have more and more resources out there. We have a European network of living labs that are able to do this. We have frameworks, the PICORI, the GRIP frameworks, Mullen frameworks, that help us to do this. So we have to commit in doing it, we have to partner with the right partners, and then we have to have the willing to create these stakeholder boards, to recruit them, to make sure that they are diverse, so we have a good representation of the patients, to make sure that we train them. And we don't want the patients, so we have a scientist with us. We want to be able to elicit the patient experience. And then we have to do our workshops, our qualitative work to be able to elicit the patient experience. And of course, finally, we have to evaluate, always evaluate how we are doing this.
Thank you very much. Thank you. Thank you, Ines, for sharing with us your insights and ambitions. What about research and development? What are the challenges you face regarding this? That's what we are going to discover right now.
Developing new drugs is a complex process filled with many challenges. A lack of diversity in clinical trials limits our understanding of how diseases affect different populations. Optimizing companion diagnostic tests for targeted cancer therapies has become an important component of personalized cancer care. So how can we overcome these challenges in drug development?
Hello, Dr. Susan Pandya. Thank you for being with us. You are Vice President of Clinical Development and Global Head of Cancer Metabolism Development in Oncology and Immuno-Oncology at Servier Pharmaceutical. In cancer drug development, how can we overcome these challenges in R&D? Please, can you explain to us how you deal with these issues?
Thank you, everybody. It's a pleasure to be here. And I'm very grateful to be a part of this privileged, esteemed panel of scientific experts. So for us, when we think about increasing patient diversity as well as patient access, I mean, these are long-standing challenges that our industry has faced. However, there are very clear opportunities we can take to improve upon these challenges. The first being our implementation of decentralized processes in the way in which we conduct clinical research. This was born out of the COVID-19 pandemic. As many of you know, our industry was really brought to our knees and had to really think creatively about how can we maintain continuity of the research efforts that are underway while still maximizing patient safety. Many of those lessons have held true and are ways in which we can now conduct research moving forward. And what I mean by that is through decentralized processes, you can make different access regarding the study available to patients who may not have ready access to tertiary medical centers. This includes remote consenting procedures, local laboratory assessments, and even potentially having telehealth visits with experts. I think this is a very rational and reasonable way by which we can tap into patient communities who otherwise would not have ready access due to logistical barriers. The second component that I think is also really relevant is our usage of real-world evidence and data. As we know, in many of the cancer settings that we are researching, there is not a plethora of information that's already readily available. And therefore, some of our hypotheses moving into clinical research can be restricted based on maybe limited factual evidence that's been published. Our mining of these data sources, I think, will allow for us to have a more descriptive understanding of patient outcomes as we prepare to go into these rare cancer spaces, so we can either enrich for those populations or consider how we want to analyze their data separately. And the third, which is very near and dear to my heart, is forging strong and lasting relationships with patient advocacy organizations. This is absolutely critical to the work that we conduct here at Servier and has proved to be highly efficacious in being able to reach out to patient communities who may not have the same level of knowledge about the clinical research space, what efforts we're making for specific types of cancers, and it also serves as a two-way street for patients to educate us about what their experience is, and how can we incorporate their insights into the way in which we're designing our protocols and evaluating relevant endpoints. Now, to touch briefly upon the challenges with companion diagnostic development and patient access, this is a complicated space. As many of you know, developing precision medicine approaches requires having a validated test, and this is a regulatory requirement and makes sense for patient selection purposes. But what we also know is that there are several panels that are already available to patients in the clinic in different communities with overlapping features on actionable targets. So this puts patients in a very challenging situation when they're considering their enrollment to critical trials, where they may already know their mutational status for a given targeted precision medicine approach, but yet have to undergo additional procedures and biopsies for the purpose of a companion diagnostic test. I think our aspiration as a group, and certainly as a scientific community, would be to consider the development of a universal diagnostic with validated targets that are already known to be oncogenic drivers that can be paired with precision medicine approaches. And this could be applied at the time of diagnosis, so earlier at the institution of one's patient journey and one's therapeutic journey. And I think that would also improve access to clinical trials as well if patients know upfront what their mutational status is rather than having to go through serial procedures. Thank you.
Thank you so much. Thank you very much, Susan. Targeted therapies are a major advance, a major therapeutic advance, but they face the problem of therapeutic resistance. Let's watch another video in order to better understand.
In oncology, tumor immune escape is a major challenge. Some cancers develop resistance mechanisms that can result from genetic mutations. When cancers become resistant, treatment options narrow, leading to relapses and lower survival rates. Targeted therapies can lose their effectiveness, posing a significant challenge for oncologists. So, how can we adapt therapies to bypass resistance mechanisms and improve patients' outcomes?
Hello, Professor Hsin-An Hou.
Hello.
Hello, thank you for being with us. You work at the National Taiwan University. You are a medical practitioner at the Department of Internal Medicine at the University Hospital. Professor Hou, in your opinion, how can we overcome drug resistance with precision medicine?
Good afternoon, everyone. It's my pleasure to attend this summit. Even the patient with the same cancer is clinical and genetical and prognostic different. I think the drug resistance continues to be to the principal limiting factor to achieve cure in patients with cancer. However, the underlying etiology of the cancer drug resistance is very complex and very multifactorial, including drug resistance, the reduced intracellular drug concentration, dysregulation of a tumor microenvironment, and activation of other compensatory signal pathway. Unlike the traditional one-size-fits-all protocol, the precision medicine in oncology uses the comprehensive genomic and proteomic profiling to identify the genetic alteration and the cancer biomarker in a cancer's tumor. In clinical practice, the gene panel of a particular set of a gene that are associated with a particular tumor type could offer a quicker and cost-effective alternative compared to whole genome and whole exome sequence. An ideal cancer biomarker should be cheaper, robust, translatable, and could guide the disease diagnosis, risk assessment, prognosis, and response to therapy. I think the integrative concept of a hallmark of cancer is aimed to explore the target therapy, immunotherapy, monoclonal antibody, and a potentially cancer vaccine to treat the patient with the cancer. We hope the right drug could be given to the right patient with the right dose and right time. So finally, I would like to conclude that cancer precision medicine in oncology could improve the efficacy, reduce the unnecessary side effects, and to enhance the overall life quality of patients in the near future. Thank you.
Thank you. Thank you very much, Professor Hou. It's time now for the last video in order to better understand one of the major issues in oncology in more depth, cancer mutations. Let's watch it now.
Certain mutations can predispose some people to cancer, and the evolutionary mechanism is often poorly understood. Some cancers at advanced stages do not respond to any existing therapies. What therapeutic or preventive solutions can be developed?
Hello, Professor Wei. Thank you for being with us. You lead clinical trials in acute myeloid leukemia at the Peter MacCallum Cancer Center, and you supervise a leukemia research laboratory at the Walter and Eliza Hall Institute of Medical Research in Melbourne. We saw in this video right now the problems caused by cancer mutations. So how do you deal with this challenge?
Thanks, Ariane. And first of all, it's an honor and a privilege to be here. And I really hope that this circle of innovation and knowledge will become a beacon of success to patients all around the world. So congratulations to Servier for building this wonderful building. So when I was asked to do this talk, my challenge was, how do I cure leukemia in 3 minutes? Put aside all of the things that I've learned before, and if I could do things again, what would I do? So that was the challenge. I reflect on the fact that to develop a new oncology drug these days, it costs 4 billion euros. And furthermore, the cost of health in France, I gather, is 12% of GDP, and in the USA it's 17%. So this does not leave much room to develop and innovate in the future, and so we really need to change our mindset, I think. With leukemia, which is the disease I treat, there have been 10 new drugs approved since 2017. Ivosidenib is obviously the best of all of those drugs. In reality, it actually has the widest absolute improvement in survival. However, all of the other drugs, if you look at the 4-year absolute benefit in survival, it's about 7 to 15%. So the majority of patients, despite all of this therapeutic innovation, all of this funding, all of these resources, most patients are still relapsing. So is there a different way forward? The problem is that by the time a leukemia presents into the clinic, the leukemia has already had years and sometimes decades of accumulated mutations. And each individual leukemia may have hundreds, thousands, and possibly even more resistance mutations. And so therefore, it's not surprising that most leukemias eventually fail therapy. So in order to develop a new strategy, maybe we should be thinking, let's not treat leukemia in the most advanced stages of disease, but can we make an absolute paradigm change? Can we prevent leukemia? And how will we do this? The first observation is that we already have the tools to detect mutations which are critical for leukemia months to years before presentation, but we don't use this knowledge. The second thing is we know that many key mutations in leukemia are highly expressed in the tumour. And this peptide is an abnormal peptide which is not present in normal humans. Therefore, theoretically, it should be possible to reprogram the immune system to specifically target this abnormal protein. And furthermore, we know which types of patients are at high risk of this situation. These are patients with solid cancer, they've had chemotherapy or radiotherapy, and for instance, there's a p53 mutation present in the blood. If we watch these patients and we watch that p53 mutation grow, these patients are going to get leukemia. But at the moment, we do nothing except wait for that to happen. And furthermore, we actually have the therapeutic weapons to make a difference today, I believe, and that is in the form of personalized therapeutic vaccines. Similar to the COVID vaccine, we can potentially put 32 vaccine targets in one product, concatenated together into one vaccine, which could then cover most of the key mutations which are driving leukemia. We could identify these patients, vaccinate them, and hopefully eradicate the tumour and stop leukemia even occurring. Now, this may not happen with the first mRNA vaccine. We may need dendritic cells, we may need adjuvants, we may need additional checkpoints, but I think the model of therapy and the strategy is the right one, because what we don't want to do is to incrementally improve survival by 6 to 8 months for the next 30 years. We really need to start curing the patients, because I think this is what we need health economically, and I think this is what our patients want. Thank you.
Thank you so much. Thank you very much to all of you. I do think that thanks to your explanations, we better understand the challenges you face in your patient journey on a daily basis. I suggest we move to an interactive discussions on these topics. Now we'll have an overview on healthcare system challenges and enhancements. Let's start with you, Hans. We have seen that it is possible to involve patients in the development of new therapies with patient co-researchers. Are there other ways of involving patients?
Yeah, I think yes, it works. Yeah, I think there are many ways to do so. Important things to think about is that when you really want to include patients or patient group, include them in an early stage. We often see that patients are included in a very late stage. The whole report is already ready. We want to have a patient view as well. Send it to them. And then they have about a few days, hopefully a week, to give an opinion. That's not very meaningful input mostly. So if you really want meaningful input, include them in an early stage so they can walk with you and understand the real issues they can comment on. The other part is, I think, that if you want to include patients, then contact a patient community, not an individual patient you know, because an individual patient you know will give his or her own perspective, and a patient community and a patient group, well, they are assigned to represent a whole group, and to do so, and you stimulate them to do so as well. So get in touch with a group, a patient community, and they will find the right patients to help you with the patient perspective in your project.
I think that what you have just said is very interesting, because it's important to consider the patient's needs in the development of the treatment pathway, of course, but also in the patient journey. Ines, what else do you think could be done today in order to improve the patient journey using digital technology?
So we already know in a clinical perspective that there are tools out there that are approved and that can make a difference. So we have several trials focused on different patient populations that have demonstrated that when we collect patient-reported outcomes and we have alerts associated with these patient-reported outcomes and a coordination with the care team, we improve patient experience, we improve quality of life, and we can even improve survival. This was demonstrated by the START trial, PROTECT, CAPRI, eSMART, eRapid, just to give you an example. So first thing, there are tools out there, including patient remote monitoring, where you can capture the patient experience, act on that, improve patient experience. There are also digital therapeutics out there that are focused on mitigation of symptoms that have proven efficacy in the care journey. So you have several digital therapeutics that are focused on improving symptoms like insomnia, fatigue, hot flashes, sexual dysfunction, cognitive dysfunction. So these are examples of digital tools that are available, that are proven efficacy, and that can improve patient journey, and we can use in clinic. On a research perspective, I think more and more our experiments have to capture patient-generated data, capture patient-reported outcomes, being able to integrate the good patient-reported outcomes, the good instruments, capturing also passive data, including wearables, commercial wearables, newer generation of wearables, collecting qualitative data. So we know, for example, we have a cohort of cancer survivors, the CANTO cohort, where we collect all this data, and we have been learning a lot of how can we improve the patient experience, because we have this data and we nested interventions using this data.
Thank you so much. Professor Hou, Ines was just mentioning the role of digital tools in order to gather data on the patient's experience. Do you think that multi-collaboration between doctors, governments, pharmaceutical laboratories is a key solution?
Yes, I think the answer is yes. If you want to go faster, go alone. If you want to go further, go together. I think this principle also applies to patient care in oncology field. The active collaboration among government, spectator, industrial, medical professional, and patients are very important to foster the patient care. Traditionally, we apply to disease center treatment. However, now and in the near future, it's very important to apply data-driven patient-centered treatment to get the drug availability and to give a high quality of patient care. I think this is very important to get collaboration among each other.
Thank you so much. Professor Wei, you mentioned in your speech earlier efficiency, and now that's what we're talking about. We're talking about how to work more efficiently, yet we must keep in mind the fact that current therapy and trial efforts are often focused on late-stage disease, and currently the practice does not effectively treat certain cancer mutations. So in order to address this, you suggested implementing a preventive strategy. The question is, do you think that all health systems in the world can sustain this strategy?
Obviously, applying to the whole world is always a big high bar, but if we think about it, the current therapies we have and develop at the moment are not accessible in many parts of the world. I went to Swaziland with my daughter last year, and in Swaziland, 1.2 million people, they have one oncologist. They've spent the last 20 years trying to solve the HIV revolution, but as people are now getting older, they're now about to be hit by a cancer revolution, which they're completely unprepared for. They can't even give patients imatinib at the moment. And so treating patients who are advanced stage, sick, needing lots of resources is not going to be a solution for the third world, but it's also going to be a challenge in the first world, because our hospitals right at the moment are filled to the brim. Everyone is struggling for beds, and all these new therapies which require a lot of supportive care I don't think is sustainable. So we must move to some kind of preventive strategy across the cancer spectrum. Vaccine therapy is one option. I think that's great, but I think our mindset has to be, how do we stop cancer from occurring earlier and keeping people well, rather than waiting for them to develop the disease and then trying to salvage a situation where we're battling thousands of mutations, drug resistance, patient complications, risk of infection, concomitant drug interactions, etc.
Thank you. Dr. Pandya, as we have seen, the resistance to cancer is a real problem, and of course a prevention strategy could be a solution, but what could we do in order to overcome these resistance mechanisms and in order to consider them from the outset of clinical trials?
Yeah, that's a great question. I think it builds a lot off of what's been discussed by Dr. Kalin and Dr. Wei as well. I think when we think about the research efforts that we're doing, at least in the phase 1 context, it's really important to plan to collect important information to understand the genetic evolution of the disease once a therapeutic intervention has been applied. Historically, we relied heavily on archived tumor samples and, sparse collection on study of biopsies for a variety of reasons. Patients may not want to submit to a biopsy, or it might be in a difficult to reach location. What we're trying to do now is incorporate early measures of evaluation of how the DNA of the cell or the biology is shifting with the application of therapies, and that can be done through circulating tumor DNA assessments longitudinally, paired with biopsy assessments that can be performed. And then I think having reverse translational exercises underway where we collaborate with our research colleagues as well as our external scientific colleagues in understanding what pathways potentially are being altered in association with the intervention, or what pathways were already altered and therefore the patient was unable to respond appropriately to the targeted intervention. Being able to integrate those pieces of information and then develop predictive models in the preclinical space, I think would then allow for us to develop adaptive trials whereby we can explore combinations up front or even through a sequential fashion. So a lot of work to be done, but I think that it's plausible given all of the technology that we have in front of us today.
Thank you so much. So it's now time for the second topic of this panel, concrete solutions for the medicine of tomorrow. We will now consider a future of medicine that is significantly different from today's research therapies, with major advancements and innovation that will revolutionize patient care. According to you, Hans, the development and the regulating pathway for a treatment should be focused on unmet needs in a patient group. And if I understood properly what you think, you think that overall survival is too limited as a single focus for measuring the value of a treatment. So what do you mean exactly, and what could we do in order to improve patients' quality of life?
Well, that's indeed a development we all see, I think. For example, I take the example of multiple myeloma again. When you only regulators and health technology assessment often do, they look at the overall survival. But looking at all those treatments before that, try to figure out which one gives the value to overall survival and which influence and which factor, etc. So when you have all those drugs and combinations, that would be difficult. You cannot figure out what the value of a drug is without other surrogate endpoints, I think. That's, for example, the MRD, the depth of remission, the PFS, and normally a deep remission will lead to a longer PFS, and a longer PFS will lead to overall survival. So a combination is important. But still, and especially health technology assessment regulators say that's not always true, and that's they're right in that. For example, when you have a very deep remission, but it affects very much your quality of life as a patient, because there are a lot of side effects, you won't get to a long PFS probably. So that's why we always say combination, not only overall survival, PFS, depth of remission, but combine it with PROs. Well, then, because then you have really all those endpoints that say something about the real value of a treatment. And we need to go that way, I think.
Thank you. Dr. Pandya, for sure, tomorrow's medicine and clinical studies will be accelerated by by new technologies. In your opinion, well, I mean, everyone talks about artificial intelligence, but in your opinion, what role will artificial intelligence play in the development of new therapeutic solutions?
Yeah, that's a loaded question. Well, we know that artificial intelligence is ubiquitous in society, right? It's pretty much everywhere and touches every facet of our day-to-day lives. In drug development, it exists as well, and I would envision in the future it would be a part of every aspect of how we develop therapies, starting from preclinical target discovery through clinical exercises, as well as the incorporation of novel endpoints, such as the integration of health-related quality of life, as well as safety endpoints to help us ascertain the overall benefit risk of a therapeutic product. Oftentimes we're relying on hard endpoints that are objective in measures such as progression-free survival, overall survival, response rate data. So I do see the utility of using artificial intelligence to hopefully be able to mine the data in such a way and learn it in such a way that allows for us to have a direct application and speed the delivery of therapeutic innovation. Here at Servier, we have several work streams in play, and I think the challenge for us will be how do we integrate this in a cohesive fashion such that we can accelerate our discoveries as well as our application in the clinic at a faster pace.
Talking about new technology, Professor Hou, maybe we could talk about new generation sequencing. As far as precision medicine in oncology is concerned, new generation sequencing is a way to adapt treatments to each patient. You have been working a lot on this issue. Can you please tell us more about it?
Yes, I think it's very clinical relevant to apply the mutation-driven or molecule-driven in diagnosis and also in treatment and also in disease monitoring. By the way, if we have both data, one is from clinical trial setting and another is from real-world evidence. And another important issue, I think we can use such data to make a comparison between the Western and Eastern countries, because some disease has a pharmacogenetic impact. All this together, if we can apply the most modern NGS-based diagnostic treatment and monitor, it could be cost-effective now and in the near future. So I think this is a very important point.
Thank you very much. Ines, meeting patient needs is something very important, and that is what Hans was mentioning earlier. Today we are evolving towards more personalized medicine and towards more input from patients. How can digital healthcare help us deliver better personalized medicine that really meets patients' needs?
So I think we are in an era where digital technology is there. It's in all domains of society, and particularly in health can be a vehicle to help us to deliver a more personalized, a more participatory, a more preventive type of healthcare. So if I think about a roadmap of how digital health can help us to deliver a easier healthcare, I would think first it can be an integrator. It can be a vehicle to integrate standardized data, multimodal data, clinical, patient-generated data, omics data, imaging data, tumor data, that will allow us, thanks to modeling, including AI, to better understand and to better stratify the patient that is in front of us, not only on a tumor perspective, but also on a host perspective. What is the likelihood that this patient will have a long-term toxicity? So first, an integrator. Then also, digital technology can help us to assist decisions. So we now have several solutions of tumor board integrated in electronic medical records that speed out tumor board processes, that reduce errors. So we can imagine that all this integrated data, if we can input intelligent and assist doctors to have the good evidence in the good moment, we will be able to accelerate decisions and to make more decisions. And then digital health can be a good tool, digital technologies, to coordinate better care, to allow everyone that is in the pathway of the patient to communicate better, coordination of care, role delegation. And as we mentioned previously, health can be the solution to empower patients, give them more education, to give them access to self-management support. And as Susan mentioned, can be the vehicle to deliver digitally enabled clinical trials that can reach more people and can democratize access to trials and also to care.
Thank you so much. Professor Wei, there is an old English proverb that says, an apple a day keeps the doctor away, yet very often it's much more complicated, of course, than just eating an apple. Preventing is sometimes easier than healing. Could you please tell us more about therapeutic vaccines?
So at the moment it's still in the very early stages. I believe there was a very interesting study from Moderna where they have shown the possibility of personalised therapeutic vaccines having potential. I think that's really excited the field about this possibility. But I think there's many questions we need to address, and that is what's the abnormality of the immune system first that allows that patient to develop leukemia or some other cancer. And for me, it's always been a question, is the patient's immune system already predisposed to getting that cancer, or is it something to do with the mutations within the tumour that is paralysing the immune system? So I think that is a really fundamental question that we need to address. We're doing active research on this, particularly with respect to P53, and I think understanding the biology of the immune system, particularly in the pre-cancer situation, we need to know more about this, and we need to really address this in order to make sure that our therapeutic actions are going to be successful. So I think it's going to be a challenging and a long road, but I think sequencing, using that sequencing information to program personalized vaccines, at the moment seems like a bit of a dream, but maybe in 10 years technology will advance very quickly.
Thank you so much for your bright answers. Thank you.